Good afternoon, everyone, and thank you for being here and listening to my talk and learning about my research. I'm just gonna start sharing. While I figure this out, I'm a postdoc. I'm a fifth-year postdoc in Dr. Deepak Nagrath's lab in the Biomedical Engineering department. I received my PhD from Rice University in 2016, and I moved with Dr. Nagrath here in the beginning of 2017. And most of my work, my research has been focused on developing and optimizing both empirical and computational tools to help us understand cancer metabolism and trying to discover therapeutic targets, particularly metabolic targets for cancers. A lot of our work has really been focused on looking at cancer or solid tumors as complex ecosystems that do not just have cancer cells that exist in a vacuum, but components that continually interact with each other. A lot of these interactions are critical for the progression of cancers, for progression of tumors. And really, we're trying to build tools, analytical tools that can help us quantitate and understand these interactions so that we can utilize them for therapeutic benefit. For the audience that is not very familiar with cancer, I'll start off with a very simplistic explanation of it. So healthy cells can acquire genetic mutations that can lead to downstream interactions in biological networks that lead to emergence of phenotypic traits in healthy cells that lead to their transformation into cancerous cells. And these phenotypic traits are commonly known as the hallmarks of cancer. Some of them you may know of. Cancer cells have the ability to replicate uncontrollably and in an unlimited fashion. They're able to resist programmed cell death, and they have this ability to invade the lymphatic and the vascular system to metastasize to other organs. For us as a lab, we understand cellular metabolism, and we think that it lies at the heart of all of these different transformations. These genetic mutations and altered biological interactions in cancer cells and also environmental factors dysregulate cancer metabolism and in essence facilitate a lot of these hallmarks of cancer. So the metabolism of the cell is basically its engine. It utilizes food and nutrients, breaks it down so that the cell can produce energy, produce simpler molecules for performing different biological function, and whatever it doesn't use is expended as heat or waste. And the cellular metabolism is a very tightly regulated process and also complex. As of now, we've documented over 13,000 reactions that happen across 4,000 metabolites. And the metabolic network looks something like this. So a lot of the work on cancer metabolism has really focused on a very small portion of this, the central carbon metabolism, which is responsible for the energy metabolism of cells. And a very good example of metabolic dysfunction in cancer cells has been the Warburg effect, which was characterized about 100 years ago, where Dr. Warburg observed that cancer cells, even in the presence of oxygen, tend to utilize glucose, convert most of it into lactate, and produce ATP in a very inefficient fashion. Whereas healthy cells would typically, in the presence of oxygen, utilize glucose and produce ATP through oxidative phosphorylation, which is a very efficient process. And it's metabolic traits like these that distinguish cancer cells from healthy cells that can really help us understand very critical differences in cancer cells, and we can use them to our advantage to find targets that can selectively kill off cancer cells and not affect healthy cells as much. And a lot of our motivation has been the fact, or rather the question that how can we quantitatively study cancer metabolism to really effectively understand these metabolic differences between cancer cells and healthy cells. And we have a lot of tools at our disposal. We have metabolomics techniques that help us measure metabolite abundances within these cells. We can also measure what's happening outside these cells, where we can effectively measure the consumption and secretion of metabolites by cancer cells. And we have the ability to consolidate all of this data, use high dimensional data analysis techniques that we have at our disposal to sort of put together a metabolic picture that represents metabolic traits of cancers. But really, one of the most important tools that we have at our disposal is metabolic flux analysis, where we can essentially measure the intracellular metabolic activity of cells. And, and metabolic, the metabolic physiology of these cells are actually directly represented by metabolic fluxes, or the rate of conversion of these nutrients and metabolites through the cells, essentially. And these fluxes are a direct representation of cellular physiology and their metabolism. And it's very akin to looking at the flow of traffic rather than individual vehicles on the road, where we can understand that some roadways are more important than the others. In the same way, we know that some metabolic pathways in cancers are more important than other metabolic pathways, which can then later be targeted for therapeutic benefit. And understanding or actually quantifying these intracellular fluxes can help us understand cancer metabolism. Unfortunately, most of these intracellular fluxes are not directly measurable with empirical techniques. So we rely on mathematical tools, particularly first principle models like mass balance, where we can translate the metabolic network of a cell into mathematical equations, use the pseudo steady state assumption to describe what's happening inside the cells, as long as we can measure what's happening outside the cell. But these flux analysis techniques, this metabolic flux analysis technique, essentially is useful for simpler toy models, simpler mathematical models, where we're limited with the predictive ability of these models to the number of measurements we can really make outside the cell. But we have at our disposal other empirical tools as well, particularly our ability to use stable isotope nutrients, which can help us track metabolism inside the cell as well. Generally, what we do in these stable isotope tracer experiments is that we replace commonly found nutrients in cell culture media with nutrients that are labeled with heavier isotopes. In this example, I've shown 13 carbon glucose, where all 6 carbons in the glucose molecules are replaced with a stable isotope 13 carbon, which is 1 atomic mass unit heavier than naturally abundant 12 carbon in glucose. And as we replace these nutrients in the culture media with the heavier isotopes, we can essentially trace the flow of glucose, one of the major nutrients for cells, downstream into their downstream metabolites. What essentially ends up happening is that it's like adding a tracer to a complex network of pipes connected with each other with different flow rates. And the tracer would essentially leave behind a footprint depending on the flow rates of these pipes. And the distribution pattern of these heavier isotopes in downstream metabolites can be measured with instruments like mass spectrometers. But this unique footprint that these tracers leave behind really rely on 2 things. One is the tracer that we use in these experiments and the intracellular fluxes. that essentially define cellular metabolism. And we also have the ability to really mathematically represent the flow of these tracers within cellular metabolism, since we know a lot of these atomic transitions that happen within metabolic reactions, and we can represent all of those mathematically. In addition to that simple metabolic flux analysis system of equations I showed earlier, we can add on this complexity that we have with the stable isotope tracing experiments, and create more complex models that can help us improve the predictive capability of and reliability of these metabolic models in essence. So on one hand, we can make these empirical measurements, extracellular fluxes or metabolic consumption and secretion that happens outside the cell. We can measure the footprint that's left behind with the stable isotope tracers inside the cell. And we can put all of this data into our metabolic model that is defined by these systems of equations and come up with prediction of intercellular fluxes in the cell. And to begin with, we use this tool to understand the metabolism of tumor stroma. Stroma, which is basically components of solid tumors that are non-malignant. And the tumor microenvironment, as we understand it, is a complex ecosystem with different types of cells, mostly fibroblasts, adipocytes, along with the malignant cells that exist in there, and immune cells that also exist, with that smattering of blood vessels that exist in solid tumors. And metabolic alterations in cancer cells are not cell autonomous. They really are defined by the biological alterations within the cancer cells, but also the tumor microenvironment, which is formed of all of these non-malignant cells. And we now know that the non-malignant cells in the tumor microenvironment really can support the metabolic demand and the bioenergetic demand of cancer cells to help tumor progression and survival. And over the past few decades, research has shown that cancer cells really can adapt to their tumor microenvironment and come up with ways to alter their own metabolism to be able to utilize nutrients they rely on, and sort of and also survive in the very harsh microenvironment that they really thrive in. And an important part of these metabolic adaptations has been direct interactions with other non-malignant cells in the tumor microenvironment. So for us, the question changed a little bit. It became, how can we quantitatively study tumor metabolism and use that to guide the treatment of cancer? And one of the first components, or one of the first mechanisms we looked at was direct metabolic exchange between stromal cells or non-malignant cells and cancer cells. And we had done an extensive amount of experiments on ovarian cancer cells. And we understood that cancer-associated fibroblasts were an important component of ovarian tumors. They're typically the most abundant non-malignant cells in these ovarian tumors, and these CAFs are essentially transformed from normal ovarian fibroblasts during tumorigenesis. What we knew before this was that some ovarian cancer cells rely heavily on this nutrient, on this amino acid glutamine for proliferation and survival. They can essentially not survive without glutamine in the culture media. However, when we co-cultured these cancer cells, these glutamine-dependent cancer cells with CAFs derived from ovarian tumors, they essentially stopped caring about glutamine present in the media. However, this effect was not observable when we co-cultured them with normal ovarian fibroblasts. So there was something special about these CAFs that essentially helped them support the glutamine requirement of these cancer cells. And bioinformatics analysis helped us understand that, or at least indicated to us that these CAFs particularly had the ability to anabolize or synthesize glutamine in some fashion, and that was probably responsible for supporting the glutamine requirement of these ovarian cancer cells. Uh, a few other interesting observations we made was that normal ovarian fibroblasts themselves were very reliant on glutamine for their growth, for their survival and viability. Um, and calves were essentially, they were agnostic to the presence of glutamine in the culture media. But more interestingly, calves were able to secrete glutamine in culture media deprived of glutamine. So there was definitely something going on. There was an altered metabolic pattern in these calves that allowed them to synthesize glutamine from the nutrients that were available to them. And what we did was we utilized our 13 carbon metabolic flux analysis technique to understand what was happening in normal ovarian fibroblasts as well as cancer associated fibroblasts to really understand how they differed from normal fibroblasts and were able to produce glutamine. So in normal fibroblasts, what we realized was that if we deprived them of glutamine, they were unable to support a critical component of their central carbon metabolism, which was the Krebs cycle or the TCA cycle, which is coupled to their oxidative phosphorylation. They were, in the absence of glutamine, they were unable to really adapt to that absence of glutamine through alternative ways and essentially had very poor viability. However, calves, on the other hand, were really prolific in adapting to that glutamine absence. And they were able to utilize glucose to compensate for whatever lack of glutamine they had in the culture media to drive that TCA cycle requirement that they have. But they were also able to use aspartate and asparagine as sources for carbon and nitrogen to synthesize glutamine that they were then secreting, which were essentially being used up by these cancer cells. So this way we were able to use this technique to understand what was happening outside the cancer cells in the tumor stroma, particularly these cancer-associated fibroblasts, and how they were supporting the metabolic needs of these ovarian cancer cells. Another metabolic interaction, a type of metabolic interaction that we looked at was through these extracellular vesicles that are secreted essentially by all types of cells, but particularly in these solid tumors that we were looking at. We found that these exosomes, which are a particular class of extracellular vesicles secreted by cells, in this case, exosomes secreted by stromal cells, non-malignant cells, they were able to support cancer cells that existed in metabolically deprived culture medium. And what we realized was that these CAFs were able to package, we essentially discovered this in the lab, that CAFs, these cancer-associated fibroblasts in prostate and pancreatic cancer cells, were able to package free metabolites into these exosomes, which they were then secreting. And those exosomes were being taken up by these cancer cells to essentially compensate or rather adapt to the metabolically deprived culture medium they were in. And this sort of physiologically represented a solid tumor where they do exist in a nutrient deprived environment. In vitro, we were able to see this happen at a functional level where without certain essential amino acids like glutamine and phenylalanine, pancreatic cancer cells had poor viability, but the moment you supplemented their culture media with exosomes derived from these stromal cells, they were essentially able to regain that functionality, that viability that they lost in the nutrient deprived medium. So at this point, we knew that exosomes were able to at least compensate for that deprivation of some certain essential amino acids. We just didn't know how they were doing it and by how much they were able to do it. So we designed this experimental system where we were able to trace metabolites that were being packaged by stromal cells or calves. In these exosomes, we characterized the secreted exosomes themselves, and we fed those labeled exosomes to these cancer cells to see how the metabolites within these exosomes were being used up by the cancer cells to support their metabolic needs. And then along with this experimental design, we essentially developed this metabolic flux analysis technique called exosome mediated metabolic flux analysis, which could help us quantitate that packaging that happens at the forefront of this system of metabolites being packaged into exosomes, those exosomes being internalized by cancer cells and then using them for their own metabolic function. So on the left-hand side of this equation were the cancer-associated fibroblasts, where they were using whatever nutrients were available to them to support their own metabolism, but also dynamically packaging those metabolites into exosomes. And with this ExoMFA, we were able to quantify how much of the metabolites from the calf's own metabolism were being packaged into these exosomes. And then on the other hand, we were able to, feed those exosomes and then quantitate how much of those metabolites were being used up by the cancer cells. And we had to do this dynamically. So since we had only a limited number of exosomes to feed to these cancer cells and they were really reliant on these exosomes for their viability, we looked at how the cancer cell metabolism changed when they were exposed to these exosomes. So in the first 3 hours, cancer cells were pretty viable. They were using about 30%, they were supporting 30% of their TCA cycle fluxes with the metabolites that they received from these exosomes. However, as time went on and the exosomes present in the media were being continuously internalized by these cancer cells, that support, that metabolic support for their TCA cycle they were receiving from their exosomes decreased slightly. And then by 12 hours, that had significantly reduced quite a lot. What we'd also seen experimentally later on, that if we were able to replenish the culture media with more exosomes, this cycle reset itself. And then the cancer cells were able to use up metabolites from these exosomes again. And the experimental design, since it was a batch process, it really doesn't capture what's happening physiologically in these tumors. But at least we were able to see what was happening in, or we were able to find a good enough representation to see what would happen in tumors in reality, where these exosomes were really supporting these nutrient-deprived cancer cells. One important byproduct of this algorithm was really being able to predict the rate of internalization of exosomes by cancer cells, which was proving really difficult doing experimentally. There weren't a lot of good techniques available to quantitate the rate at which cancer cells are able to internalize these exosomes and use the metabolites they have within them. But with the ExoMFA algorithm, we were even able to do that. So apart from the metabolic interaction, the metabolic support CAFs, so providing to these cancer cells, we were also able to answer this one biological question about the mechanism of uptake of exosomes by cancer cells with this algorithm. Yeah, So this sort of summarizes what we were able to do. We were able to quantify what was happening in the donor cells, which were the calves, which were packaging metabolites. We were able to quantitatively prove that the metabolites packaged within these exosomes was supporting the energy metabolism of cancer cells. And essentially what we had designed was a 13 carbon metabolic flux analysis algorithm that connected a multicellular system, which was one of the, I think, the first ones that were available at the time. And another advantage that I haven't listed here is that this algorithm can easily be adapted to any other system where there is transport of free metabolites through any type of extracellular vesicles from a donor cell to a recipient cell.